Hello again. In the second part of the video, we will focus on B cells. First, we will see what is the phenotype of cells. And then, we're going to talk about how the B cells change their phenotype while they mature, while they grow up. Before we talk about maturation, we have to understand another basic principle: how we recognize the cells in the blood. Imagine that you are a hematologist and you're asked to make a diagnosis by carefully watching the cells under the microscope. Well, when you see, let's say, this cell or this cell, is it possible to say if it's a B cell or a T cell? In other words, can we distinguish them morphologically? The answer is no. We need a more powerful tool that is called flow cytometry. We will talk in details about this tool on another video. For now, we only need to know that flow cytometer is a machine being able to detect proteins in the surface of the cells. The proteins are called clusters of differentiation, CDs for short. What does it mean? CDs are antigens, meaning they are proteins that are used as cell markers. So in the beginning we thought that they are present only on the surface and only on leukocytes. With years, things have changed, and now we know that there are some intracellular proteins as well, and they can be present on other cells than leukocytes, but we will focus here on leukocytes. Clusters of differentiation can act in numerous ways. Let's see some of them. Often they act as receptors, transmitting signals to the interior of the cell and change the behavior of the cell. Other times they act as ligand proteins that help cells connect to each other. There are more than 400 CDs, and scientists over the years had meetings and discussion to agree what the names of the CDs will be. So for most of them, we use numbers such as CD19, CD20, CD40, etc. For others, they did not use numbers, for example this one, HLA-DR, found on monocytes, or this one, called surface immunoglobulin, SIG, to make it short. So, why do we call it surface immunoglobulin, and what has to do with antibodies? Actually, immunoglobulins are antibodies, and they are also proteins. It's the same thing. The first antibodies made by newly formed B cells, the naïve B cells, are not secreted. They are found in the surface. Mature B cells, called plasma cells, have Igs in the inside and they are capable of secreting them. Okay, go back to the CDs now. Do I have to memorize 400 CDs to become a doctor? Of course not. Most of them we do not know what they do. However, CDs, the cell markers, are super useful in two ways: diagnosis and treatment. How so? Let's see. First one, how the CDs are useful for diagnosis. The different combinations of CDs create different immunophenotypes. What does phenotype mean? What the cell looks like. For example, the first one looks like a normal B cell, and the other one looks like a not normal cell. Why immunophenotype? Because we are able to detect it by immunological methods. What's this? It's a method where a specific antibody is used to detect a specific antigen, like this. So let's take this example. Imagine the cell above that looks like a normal mature B cell because it has CD20, CD19 and surface IgEs that characterize the cell, so we can tell it's a B cell. The other one is the sick one. It expresses CD5, 23 and weakly IgG, which is not normal. We can see all that by microscope? Of course not. So we need the markers, the CD antigens. When we have a clinical suspicion of a hematological disease, you can ask your lab to perform a test. Put the antibodies in the sample of a patient's blood and see what happens. This test is called flow cytometry. It's a method based on principles of immunology and gives us information on cell phenotype. So we have put antibodies against CD5, 23, 19, 22, etc. And once there is a match, The fluorochrome associated to the antibodies will be excited and sends a signal to the machine. Each antibody-antigen match gives a different signal. For example, CD5 matches antibody against CD5 and gives a purple signal here, so we can tell it's a leukemic cell. Fantastic, right? Okay, an example for treatment now. Rheumatoid arthritis is an autoimmune disease that affects the joints and also other organs, lungs, skin. This happens because the antibodies attack our cells. Rituximab is a drug that we call monoclonal antibody. It targets the marker CD20 found in B cells. So the rituximab binds the CD20 and forces the B cell to die. Yeah, but also forces the normal B cells to die. This is the problem, and this is why rituximab is immunosuppressive agent. Okay, so now you're convinced that we have to remember at least some of the CDs. We're going to look at the phenotype of the cells while they mature. The final step for B cell development is the plasma cell, the cell capable of secreting specific immunoglobulins, a sort of sophisticated weapon against a specific enemy. Now we're going to see every step between the two cells. What do they look like? Okay, remember this one? All the B cells have a common ancestor, the multipotent hematopoietic stem cell. In the first video, while I introduced the B cells, we've seen that the multipotent hematopoietic stem cell looks like a multicolor lizard. It's capable of transforming to any type of cell. So this lizard will be our stem cell. I like to see like that. So let's imagine that CD34 corresponds to red and CD117 is the orange color. These are immature markers. Okay. Now as the stem cell becomes a lymphoid progenitor, it starts having properties of lymphocytes. This cell is called now a pro-B cell. Pro-B cell. B cells are characterized by line-specific antigens, CD19, I will color it in light blue, and CD22, it's the green one. So CD19 and CD22 are B cell markers. Pro-B cells express also CD10, another marker associated with maturity. It's brown, so the pro-B cell would look like this. Very beautiful. Next step of the development is the pre-B cell. Now, as the B cell develop to more mature forms, they keep their distinct markers of the B cell line and progressively they lose the CD10, the brown color. And they get another characteristic marker of the B cell line, the CD20. Pre-B cells also start to develop proteins in the inside, very light blue. Very important to remember all that. Now the next level of the B cell is to become a naïve B cell. Why we call it naïve? Because it's not yet in contact with antigens. The naïve B cell expresses the B markers CD19, 22, and 20, and it starts expressing surface immunoglobulins that are called IgM. Electric blue. This cell has to pass an important step now. Survive apoptosis in the bone marrow. So if the cell survives apoptosis, it migrates to the follicles of the spleen and the lymph nodes, where the cells pick the antigens, aka enemies, and it will become more mature and able to fight them with specific weapons, the antibodies. So the B cell has now become a follicular circulating B cell. At this stage, it gives its B markers CD19, 22, and it starts expressing CD23 in dark purple, and another surface immunoglobulin, the IgD, in light purple, and it continues expressing IgM as well. So it looks like this. Very, very beautiful. And now, last but not least, the last step is the plasma cell. Follicular cells circulate continuously between the bone marrow and the lymphatic organs until they meet an antigen. Once they meet an antigen, they become either memory B cells or these fantastic pink plasma cells. They are a little bit particular, though, because usually They do not express strongly the B markers, although they are B cells. They express CD38 in light pink and CD138 in dark pink. So we will remember plasma cells as the lizard with no blue color, no B markers. As opposed to the B cells that have surface immunoglobulins, the IgEs of plasma cells are cytoplasmic because they are made to be secreted. Additionally, as opposed to other type of B cells that circulate in peripheral blood, they are absent in blood. Quick recap: no B markers. They live in the bone marrow. They have no surface IgEs because they're meant to be secreted. And when plasma cells are sick, they cause a disease called multiple myeloma. We will learn a lot about this disease on an upcoming video. Great. So this is the journey of the hematopoietic stem cell to become a plasma cell. He first becomes a pro-B cell, then a pre-B, then a naïve, that circulates between marrow and lymphoid organs until it meets an antigen, and it becomes either memory B cell or plasma cell. Very simple. Okay, we have to remember all that to become doctors. This was my video on the changing phenotype during B cell differentiation. Thank you for watching.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.